On a day that would otherwise be bleak, and that is the day that NHMRC grants results are released, it is an absolute pleasure to have a bright hour in the day, and that is a bright hour that is going to involve a centenary lecture given by Professor David Williams. And David, it's absolutely tremendous to have you here. As I will tell the audience, it's obvious that this year is an exceptionally busy year for you, as you are President of the American Society of Haematology, one of the largest scientific societies and clinical societies in the country, and we very much appreciate the time you've taken out of that exceptionally busy schedule to help us celebrate our centenary. So thank you so much for your generosity. David is the Chief of Haematology Oncology and Director of Clinical and Translational Research at Boston's Children's Hospital and Associate Chairman of the Department of Paediatric Oncology at Dana-Farber Cancer Institute. And David really has been the driving force in development of that haematology-oncology program in Boston. And in fact, it's the third seriously large translational research centre that he has spearheaded and developed, the others being in Indianapolis and Cincinnati. He has been doing research both at the translational end of the spectrum, and he's going to talk about some remarkable gene therapy trials that he's been carrying out over the last few years, but also made some seminal discoveries in basic haematology involving, I guess, signal transduction between stem cells and their microenvironment. I should also say, having known David for a little while, he not only is a tremendous leader of the field as evidenced by his presidency of ASH, but he's also a remarkable mentor, and we owe him a debt of gratitude for the work he did preparing Andrew Roberts, among others, for leadership here. So David, it's an absolute privilege for us to be able to welcome you to give a centenary lecture, and thank you so much.
Thank you. Thank you very much. I'm so pleased to be here. You don't have to thank me, actually. I thank you for inviting me. I don't want to dwell on the past very much, but just to say it's very special for me to be here. When I was a postdoc, which is now over 30 years ago, at MIT, and trying to learn the field of hemopoiesis, there were two places in the world that stuck out in the literature. Manchester, England, and the WEHI. And I had the great fortune early in my career to meet Don Metcalf and Mike Dexter, both of whom were exceptional in the way they were generous with their time for some young upstart that they didn't know from Adam. And it really impressed me that science was a great thing to be in, when there's great people like that would take their time to talk to a postdoc and give them advice. So that was an early teaching moment for me, and I've tried to develop my career with the same kind of care and generosity for trainees, and I hope I've had some small impact in that way. Not nearly as much as those guys, but certainly I hope some. So that's why it's so nice for me to be here, and when invited, I accepted immediately to come and visit the WEHI again. This is, I think, my third or fourth trip here. It's always wonderful to hear the science, and I had the great pleasure yesterday of being at the Royal Children's and seeing a wonderful facility there. So I've enjoyed it very much. I'm going to talk today about gene therapy and monogenic diseases, and I'll end with some sort of thoughts about how this may lead us into the next era of genome editing in the clinic. So I'll just give a quick overview of gene therapy, and I'm going to use X-linked severe combined immunodeficiency as a model disease in which we've learned from mistakes in the first round of clinical trials, going back to the laboratory, re-engineered vectors, and then went back to the clinic, and I'll show you some data from the new trial that we're continuing on. And then I'd like to spend more time on unpublished data where we're using, I think, a novel approach in gene therapy, which is to incorporate knockdown of a gene to treat, hopefully, sickle cell disease in a trial that we hope to have open in 2016. And then, as I said, I'll just end with a few thoughts about future approaches. So for those of you not in the field, just make some sort of preliminary remarks. There's sort of two broad ways that one can think about gene therapy as a medicine. One is what we call in vivo gene therapy, where the DNA is directly injected into the human being, and you can do that in multiple forms. And the success in areas like hemophilia, for instance, are an example of that. We've mainly focused on ex vivo gene therapy, and that's where one takes the target cells out of the body, takes them to the laboratory, manipulates them using gene transfer approaches, and then reinfuses the cells back into the person. And that is built, as you can imagine, I guess, on the foundation of bone marrow transplantation as a therapeutic modality. And so in this case, we remove hematopoietic stem cells from the patient, use a viral vector because of its efficiency of gene transfer, transduce some number of stem cells, and transplant these into the recipient. And depending upon the disease, then you either have full conditioning or, in some cases, no conditioning at all before the patient receives the cells. And of course, the advantages of this are clear. There's no donor search, so in diseases where time is of importance, that's a help. And you completely eliminate the risk of GVHD. And I think in the future, maybe also we can approach maybe non-obvious diseases because we can basically use the genetically manipulated hematopoietic system as a factory to deliver the transgene product, and by cross correction, potentially address, for instance, diseases of the CNS, where you wouldn't expect hematopoietic stem cells necessarily to have an impact on the disease. So we've seen a renaissance of this field in the last, I would say, 5, 6, 7 years. And we now have a number of diseases where the proof of concept has shown that this works. They're listed here. I won't go into them in detail, but there's 2 types of SCID, chronic granulomatous disease, Wiskott, metachromatic leukodystrophy, and adrenal leukodystrophy. And while these have been highly successful for the most part, you can see the asterisks demark trials where there was first-generation gamma retroviruses were used, and in all 3 of these trials, there was significant adverse events related to insertional mutagenesis and subsequent either myelodysplasia syndrome or leukemias. And there's also been success in the non-integrating vectors, and the 2 sort of classic successes that people point to are Leber's congenital amaurosis and hemophilia B. And the key here is that one is transferring the gene in a non-integrating form into tissues that don't proliferate and therefore don't require integration. So on the one hand, there's less chances of integration-induced transformation or oncogenesis. On the other hand, this may not be as permanent a correction as in an integrating vector type technology. So just to put things into perspective, I generated a modified timeline from Manuel Grez's published timeline in gene therapy to give you a sense, especially if you're young, for how long this has taken. So you can see the first gene transfer using viruses was done in the 60s. This is me as a postdoc at MIT. So in this era here, we first defined that you could use gene transfer to modify murine hematopoietic stem cells, and we thought at the time this would be translated very quickly into humans. That didn't work quite that way, and so the first real efficacy in human trials was shown in the early 2000s with the work done in Paris by Marina Cavazzano Calvo and Elaine Fisher. I know Elaine was recently here. This was followed very quickly then by insertional mutagenesis, and in the trial I'm going to talk to you about in detail, a number of children developed leukemia because of this adverse event. And now in our era, we've used new vectors that are improved, and I'm going to show you about that, to have success. And the higher efficiency gene transfer afforded by both understanding stem cell biology more and also by using a different vector platform, lentiviruses, have allowed us now to really broaden the indications because we're getting much higher efficiency of gene transfer, so we can approach diseases where the modified cells don't necessarily have a proliferative advantage or pressure in vivo. So what I'd like to talk about very quickly is a work on severe combined immunodeficiency disease. This is sort of known by many lay people as the bubble boy disease. This is David, the bubble boy in Texas, talking with his mother. He was raised for 6, 7, 8 years in a completely sterile bubble and ultimately died of viral infection when it was contaminated. And I think the ethics of that are such that we'll never see that kind of an experiment again, but that's how many people know this disease. It's a good candidate for gene therapy because it's, of course, fatal if untreated, usually by the age of 1, so it's a very severe phenotype. We've known the genes that cause this. There are a number of one for quite some time, and importantly, as I said in my introductory slide, this is a gene technology that's built on bone marrow transplantation, and so we've known for many years that you could correct this disease with an allo transplant. So what is the disease that, in this particular case, this is due to the common IL-2 gamma chain deficiency, and it's called that because you can see in the absence of this gene product, the immune system is severely hampered. The gamma chain, although it's called IL-2, you see actually participates in the signaling pathway of a number of cytokines shown here, and the absence of the gamma chain leads to a characteristic called T-B plus skid. So the NK and T cells are completely deficient in these children, and while they make B cells, the B cells are dysfunctional so that they can't respond to standard immunizations, for instance, because of the lack of T cell help. So in the first trial, 2 trials done in Europe, and one in Paris and one in London, there were 20 children enrolled, and you can see here on this graph the recovery of the T cell count on a log scale in days after infusion of gene-modified cells, and you can see quite successful reconstitution of the T cell compartment. And in fact, 18 of the 20 children in this trial were successes. We had, I was involved in this trial, but there were one patient that got a poor product and failed reconstitution in Paris and went on to a transplant, and a second patient from here in Australia who had, again, low reconstitution of gene-marked cells and didn't clear concurrent infections and went on to an allo transplant and died of complications of that transplant. So you would say overall highly efficacious approach to treating the disease. But subsequently, of course, multiple children in this trial, these 2 trials developed leukemia. So here are just tracings of, in the Paris trial, patient 4 and patient 5. And if you look at the time scale here, 3 years after infusion of the cells, their T cell numbers skyrocket. The T cells are lymphoblasts, and they have, in both these patients, a number of additional cytogenetic abnormalities that were found. But in any case, these leukemias were a direct result of insertional mutagenesis in their stem cell compartment. And interestingly enough, you can see we've put the data together here on the next few slides from both trials. 5 out of the 20 patients in the 2 trials developed leukemia. And Several of these ended up having insertional events in a gene called LMO2. This is a transcription factor that's required in early T-cell development. And you can see in these two patients, patient 4 and patient 5, the virus integrated either in the first intron or in the 5' untranslated region, and led to the inappropriate lack of turning off of this gene during T-cell development, something that Matthew McCormick here was involved with when he was in Terry Rabbitt's lab. Now, the clinical outcome of these patients are that one patient died of therapy-resistant disease, and 4 patients were treated successfully in standard ALL pediatric treatment. And in fact, LMO2 is a known de novo gene or a known gene that plays a part in some de novo pediatric ALLs. And the interesting thing about these 4 patients was that their leukemia was treated successfully, but they maintained an autograft that had the gene-corrected stem cells and common T-cell progenitors. And so they, to this day, continue to have a normal immune system. So we began to work as a collaborative transatlantic group at this time to try to address these side effects. And the intent was to develop an equally efficacious vector that was safer. And this was before really lentiviruses were on the scene. And so we elected to try to develop a safer gamma retrovirus vector. This is the previous vector, where the IL-2 receptor gamma chain is expressed off of the LTR, the long terminal repeat, with the Maloney leukemia virus enhancer marked here in red. And through a series of preclinical studies that I won't go into in any detail, we ended up developing this vector in which we've deleted the enhancer region from the LTR, and for expression of the transgene, we've inserted a cellular promoter called EF1 alpha. And so the major modifications were those, eliminating the U3 region and replacing it with EF1 alpha. And one of the reasons we ended up with this promoter is that it has very little enhancer activity in enhancer assays. This led to the development of an international trial, 5 sites. It's a phase 1/2 trial in which we were very careful to keep the same actual protocol as the previous 20 patients and simply replace the vector so that we could realistically compare the results with the previous trial. And the specific questions that we ask are, does the change in vector configuration actually lead to improved safety? Does the weaker internal promoter maintain efficacy? Because this would, of course, be quite important since the previous trial actually was efficacious. And from a biology standpoint, we were interested in whether the deletion of the U3 region would have any effects on what we saw in repopulation or reconstitution of the patient's system, hematopoietic system in vivo. So I'll take a moment to answer these questions, but this is recently published, so I'm going to go relatively quickly. So first of all, we plotted the recovery of the T-cell counts of the new trial, which is the SYN gamma C trial, in black, against the combined data from the first trial with the MFG vector. And you can see that the recovery of T-cells, while early on you might wonder, and we did, whether the kinetics was different, by the 2 time points that we had prescribed to analyze, 3 months and 6 months post-transplant, there's absolutely no difference in the reconstitution of the immune system in these 2 trials. So efficacy was maintained, and we next ask the question, how about safety? It's a little early to be completely clear about this because of the length of latency of the leukemias that developed in the first trial. So we looked at this in multiple ways. One is comparing the integration analysis of the pre-transplanted engineered product with the engrafted cells. So this is an assay that people call clonal skewing, and I'll go into that in a little bit more detail in a moment. We specifically cloned and looked at the integrations of all the available integration sites in the reconstituted patients in known lymphoid proto-oncogenes, focusing on lymphoid because that's the type of leukemias that the previous trial had. Examine in detail the integrations near genes that are associated specifically with leukemia in gene therapy trials. And we developed an assay system to look for clumping of integrations and then ask the question of where those clumps are in the genome in relationship to any gene. And finally, and in the end, the presence and absence of leukemia. So I'm going to quickly go through these data. Here is the comparison of the two trials. So we call SCID1 the trial in both Paris and London. So here's the Paris trial, here's the London trial, here's the current trial. And this is the common integration pattern of a lentivirus vector. And what you're looking at here is a heat map. And what you're looking for are, as you can see here, compared to lentivirus, changes in overall integrations in a variety of genes. So globally, what we found was the integrations in the first trial is very similar to the second trial, and this led us to the conclusion that the vector modification, including the enhancer deletion, in a global sense, didn't make any obvious difference with respect to where in the genome the vector was integrating. This is an example of the lack of clonal skewing. So this is the first patient treated in trials, the patient from Boston, is now past his 4-year follow-up. And what you're looking now here for is blue bars where there's a change in the integration pattern over months since the transplant, up to 4 years. And this heavy blue line up here, if you can't read it, is many, many, many integrations in none in any particular gene. So you can see over 4 years, a period, there doesn't appear to be any skewing and any clones coming up in any particular genes, which was quite reassuring. Next, we looked at the frequency of integration in lymphoid proto-oncogenes, and we assembled a list of 38 genes that are known to be proto-oncogenes in lymphoid cancer, and we arbitrarily set integration boundaries of 50 kilobases from the transcription start sites of those genes. And now when you compare the first trial, SCID1, with the second trial, there's a highly significant difference of integrations in near these lymphoid proto-oncogenes. And the way to think about this is that if you have integrations that are adversely and non-neutral way affecting the engraftment of the lymphoid compartment, then you would see more, you would hypothesize you would see more integrations in growth-promoting genes such as proto-oncogenes. So again, this was quite reassuring. And then the last thing before the leukemias was to look in a more unbiased way at integration clumps. So we developed with Rick Bushman's group a way of bioinformatically looking at integration clumps. And then we identified the clumps in both trials, again, in an unbiased way. And we asked the question, what do the clumps tell us about the integration sites? And interestingly enough, there was clearly clumping going on in the first trial. And really interestingly enough, those clumps prominently were in 3 oncogenes. So in LMO2, in cyclin, and in EVI1. And these 3 genes actually turn out to be the very genes that have been causative in leukemias in gene therapy trials previously, so that all made sense. So this is a little hard to read, but EVI1 is here, and the previous trial is in blue, and the current trial is in red, and if you have very good eyesight, you can see lots of blue ticks in EVI1 and in cyclin D1 and LMO2, and hardly any ticks from the current trial. So clearly, the integration pattern is much more neutral in the current trial than the previous trial. And then finally, although we're still early, this is just a plot of the incidence of leukemia and when it occurred in the first 2 trials, and the red is the current trial, so no leukemias and still early, but we're quite hopeful that this is showing us a safety pattern that's going to continue. So since we published this paper last year in the New England Journal, we've enrolled an additional 4 patients already, 3 in the US and 1 in the UK. We've enrolled now 7 out of 9 in the United States. We're almost done with the trial. And actually in January this year, the FDA granted us a orphan drug designation for this particular vector. And using this now, we're now going back to the drawing board and planning our next trial called SCID3. This will be another international trial. And now we're introducing the concept of conditioning, mild conditioning, because what we found in the first 2 trials, ours and the previous British and French trials, is while we're getting very good T-cell reconstitution, we're not getting very good B-cell reconstitution, which probably reflects the lack of stem cell engraftment in a non-conditioned recipient. So in this trial, we'll maintain this promoter because we've shown it to be safe and effective, but we'll add busulfan conditioning to patients that come into the trial. I'm going to skip through the summary for time's sake because I've said it all before, and I want to move on to unpublished data. So I'd like to talk now about a pretty neat project that we're fairly far into, where we're going to knock down the transcription factor B7A to treat sickle cell disease. So for those of you not familiar with hemoglobinopathies, this is a curve showing that when in utero we express fetal hemoglobin, gamma. When we're born, we suddenly turn this gene off and turn on adult globin, which is made of a component of beta hemoglobin. And it's on the beta hemoglobin that the sickle mutation occurs. And so you can see that when this switch occurs, this is called the hemoglobin switch, you start expressing a mutant form of the beta globin locus that causes the disease. Now, there's been millions and millions of dollars expended by the NIH and other agencies to try to understand this switch. And for many years, it was completely enigma as to how it happened. One thing that was clear, this is a paper many years ago from New England Journal, where Ora Platt looked at this survival of patients based on their expression of hemoglobin F. And so you can see patients that have low hemoglobin F have a different survival curve than patients that have high hemoglobin F. And the difference is about a decade of survival. And this correlates with those of us who take care of sickle cell patients know patients that have high F tend to have an attenuated phenotype. And this becomes important to understand that hemoglobin F is an important modulator of the sickle phenotype. So first of all, of course, as I've just said, when you're born, if you have sickle cell disease, you have primarily fetal hemoglobin, and babies that have that don't have a phenotype until their F falls below a certain level, and then they start having clinical symptoms. And there are populations in the world, particularly in Saudi Arabia and India, where they have a phenotype with high F, and they tend to have a much milder disease. And in the US, studies have shown that hemoglobin F inversely correlates with pain crisis, which is a major morbidity in the disease. And biochemically, we can understand this because the gamma globin, in part, disrupts the polymerization of hemoglobin in the cell, and that, of course, is the pathophysiologic feature of sickle cell disease. So in 2011, Vijay Sankaran, who was a graduate student in Stu Orkin's lab, used genome-wide association studies to come up with a startling finding, which is that the lymphoid transcription factor called B7A is a key component that regulates this switch. So it's a repressor of gamma globin expression. So after you're born, expression of B7A goes up in your red cells, and as that occurs, gamma expression is extinguished and beta expression then turns on. And Stu's lab subsequently shown the relevance of this in a disease model. And so he crossed the B7A knockout mouse into the Berkeley sickle cell mouse model, which is where the mouse locus, globin locus, has been replaced with the sickle human locus. And you can see in this cross, the amount of hemoglobin F here is 85%. Extraordinary expression of hemoglobin F in an adult mouse. And more importantly, in these mice, you can see that these sickle cells essentially are all gone. So the expression of BCL11, the knockdown of BCL11A knockout is associated with reinduction of hemoglobin F and a reversal of the sickle cell phenotype. But these animals have no B cells because BCL11 is an essential transcription factor for B cells. So our approach to this, as soon as we understood this from Stu's work, was to hypothesize that this was an absolutely great target for gene therapy, and we wanted to build a lentivirus vector to induce gamma expression by knockdown of BCL11A. And the really nice thing about this is it simultaneously turns off the mutant beta sickle gene. So it's a way of killing two birds with one stone, basically. So we thought this would be simple. It turned out not to be simple. And the reason for that is described in part here. So those of you that do shRNA screens, you probably know that those screening libraries are almost all set up in a lentivirus with a Pol3 promoter. Now, the problem with Pol3 promoters is that it leads to extraordinary expression of the transgene. And there's a fair amount of literature that suggests that this is toxic to cells. And so we from the beginning, envisioned that our clinical vector would need to use a Pol2-driven vector, or Pol2-driven shRNA rather than Pol3. And some of the advantages of that is that this mimics the structure of endogenous microRNAs in processing and yields lower and presumably more physiologic expression that may be better tolerated by the cell. There's some literature that it induces less interferon response. And importantly, it turns out, not something that we thought about necessarily in the beginning, using Pol2 allows us to develop a regulated vector. So in our first pass, we simply embedded the shRNA that targeted BCL11A, actually handed to us from Stu's lab, into a microRNA 223 backbone. And if you look here at the vector, so here's the lentivirus, we're using SSFV, which is your ubiquitous high-level expressing promoter, to drive this architecture here, which we call SH-MIR. And if you look at the predicted structure, you'll see that there's some slight differences here between what's predicted to be expressed from shRNA with or without the 223 hairpin. So what we found, unfortunately, and it's published, so I'm not going to go into a lot of detail, is when we simply embed an shRNA into a microRNA, it actually alters the seed sequence. And so we've come upon this because we kept finding that the shRNAs in one vector worked extremely well, but when we put them in a microRNA architecture, they worked very poorly. And so with a lot of work and a lot of RNA sequencing, we actually figured that what you really have to do is do a 4 to 5 base pair shift when you do this embedding. And when you do that, you can restore the knockdown efficiency to almost the level you see in a Pol3 base vector. So we then asked the question, in this system, can we demonstrate that there's excessive toxicity or rescue of toxicity using this approach? So here you see in a variety of cell lines, expression of the identical shRNA in a Pol3 vector, PLKO, versus a Pol2 vector, commonly used lentivirus called LEGO. And here you're seeing the number of transduced cells over time, and you can see very clearly that in all these cell lines, the expression from the Pol3 system is toxic to the cell, and this toxicity is primarily obviated by the Pol2 vector. So we went on then to test this in a translationally relevant system. So here we're using CD34 cells to be transduced with the lentivirus targeting BCL11A. And then after transduction, we do an in vitro erythroid differentiation differentiation assay. Then we collect the cells at the end of the differentiation and do biochemical assays. And so here you see very nicely that we can induce, this is the percent of fetal globin expression as a total, compared to the total globin. And in our modified, so these are the optimized vector, you can see that we're getting 50 to 60% of the total hemoglobin in these cells are now F. So this is a very nice outcome, and we thought we were on our way. And here is the HPLC assay that just confirms the RNA assay, where you can see very high in the black bar induction of fetal hemoglobin. But the problem then became when we tried to apply this system in vivo in a standard assay where we're transducing human CD34 cells and then transplanting them in xenografts. This is NSG mice. You see here that after a short engraftment period of 14 weeks, compared to an empty vector, it's called SEW, where we have good engraftment, 80%. In every case, when we looked at our knockdown vector, we saw very poor engraftment. And actually, I have to say, the first experiments were done by one of my postdocs. This happens all the time, right? And he kept saying to me, it's not working, and I kept saying, you're screwing it up somehow. So go back and do it again. So he kept doing it, and then I had another postdoc do it, and the other postdoc did it. Oh, he's right, it's not working. So then I believed him. So we then went back and said, okay, there's hints here of toxicity, which we wouldn't have predicted, and we certainly don't understand. Let's make sure it's correct. So we did competitive population assays in two ways. One is to use vectors that have two different fluorochromes and then compare knockdown versus not knockdown or scrambled. And the other was to use congenic strains. Many of you probably use these CD45-1 Boy J into Black 6. So I'm just going to summarize very quickly a lot of data. Each of these dots is a mouse recipient. And we've basically just asked the question, after full engraftment, if we look at engrafting targeting cells versus targeting vectors versus non-targeting in the same animal, that's the nice thing about these assays, it's very clear. Anytime we targeted BCL11A, there was no engraftment. So clearly, BCL11A is unexpectedly needed in an engrafting stem cell to engraft. And we saw in these animals, in a standard transplant, mouse syngeneic transplant, we saw what we would expect, which is a B cell deficiency because it's a B cell transcription factor. But we also saw then validation of what we were seeing in a xenograph, which is a relative lack of primitive LSK cells in the bone marrow after engraftment. So this confirmed the loss of gene-modified cells, both in B cells and in HSCs. And we clearly knew then we had to have an alternative strategy. As we were trying to understand the mechanism, we needed to translate a different technology. Now, one of the interesting things about this was at the very same time we were discussing with Sangamo for thalassemia a gene editing trial in which the gene editing product was a deletion of the gene, BCL11A. And we, honestly kept saying to them, you can't do this trial. This is not going to work if you pursue this trial. And so it took some time, but we finally convinced them that they should abandon that, and they have done that now. And I'll come back to that at the very end for a moment. So our alternative strategy then turns out that having converted into Pol2 system was very fortunate because now we could immediately go to lineage-specific expression of the BCL11A knockdown. And so here we used a commonly previously reported LCR vector, a locus control region vector, where we've taken the LCR hypersensitivity sites 2 and 3 from the globin locus, beta globin locus, hooked it to minimal beta promoter, and embedded our microRNA, shRNA downstream of that. And that's what the vector looks like. This just shows you, as opposed to the ubiquitous promoter hooked up to GFP, and here is erythroid differentiation assay and the commonly used markers of erythroid differentiation, GPA1 or GPA and CD71. So as cells, erythroid cells, differentiate from stem cells, they acquire CD71 and then acquire GPA. You can see the ubiquitous promoter indeed expresses in all cells, whereas our new lentivirus expresses very little in primitive cells, and then as the cells differentiate, they express. So does this now work in vivo? We've done the same sort of assay before, where we've now compared the shRNA and the LCR backbone with the same shRNA and the SSFB backbone, or ubiquitous backbone, and you can see in every case, again, the engraftment is due to the cells that are transduced with the LCR vector. And this just shows you, again, in a transplant setting where we've compared these for expression, that as a differentiation occurs, the new shRNA turns on in the terminal progenitor and precursor red cell population. So now, does this work? It avoids the cytotoxicity, but does it work? So here we've looked at transplantation of human CD34 cells transduced with the vector, allow these animals to engraft fully, then harvested the CD34 cells from the bone marrow and differentiated them into red cells in vitro. And here you can see induction of gamma globin. And here we're getting 70% of hemoglobin F in these human cells of the total beta globin, the total globin. And that's shown by HPLC again here, where the arrow shows you very high levels of hemoglobin F. And now we've gone on to look at this more carefully again in transplantation assays. And you can see after transduction and highly purified cell population, you get very nice expression as the cells differentiate. And here you see 45 to 50% of hemoglobin F in these cells in vivo. We've continued to modify the vector additionally, and here you can see both by PCR RNA analysis and HPLC, we have here now a construct that's, you can see, consistently giving us over 60% hemoglobin F in induction of CD34 cells, red cell derivative from those. So we've now taken this new vector called D12G5 and done additional studies, just quickly shown here. Here's the knockdown of BCL11A. Here's the XL form, which controls, we think, repression of the gamma locus. So you can see very effective knockdown. Here's RNA induction, RNA analysis. And here's hemoglobin F production up to 60%. And now we've gone on to look at this in sickle cell CD34 cells from, now we're up to 4 patients that we've looked at. Here again, 3 different ones are very well knocked down. And now in these sickle cell patients, 80% of the globin in the sickle red cell is hemoglobin F. So these cells, of course, can't sickle. And we've now tested this in the Berkeley mouse model, and it's a complicated slide. I'll just point out really 2 things. One, in this red bar, is the number of reticulocytes in the animal. So this is, sickle cell has a high retic count. So we can reduce the sickle baseline tick count in these animals from 40%, 35 to 40%, down to less than 10%. And you can see normalization of their other red cell indices. So this is clearly a very effective approach to reducing the hemolysis that's caused by the sickle mutation. So to summarize this before I conclude with some just final remarks, we've developed a conversion rule for transferring shRNAs into shRNA mirrors using the Pol2 promoter, which really reduces non-specific toxicity of shRNA expression, something that many people don't pay attention to because they're working in vitro systems where the toxicity may not be all that relevant. We've identified very potent shRNA mirrors that target BCL11A that mediate a very significant induction of hemoglobin F and reversal of the hemolysis of the sickle cell Berkeley model. And this erythroid-specific expression then avoids a previously unrecognized toxicity of BCL11A knockdown in stem cells, and previously unrecognized in part because the phenotype is mostly seen when you do a transplant of the cells, and we can come back to that in the question and answer period. So let me end with some thoughts about the future of gene therapy. I think what's happening in the field is that we're moving towards a more sophisticated approach to vector development where we want to ensure that expression of the transgene really in those cells where it's physiologically needed. And this is including the use of chimeric promoters. So for instance, in our currently open chronic granulomatous disease trial, we're using a very chimeric myeloid-specific promoter to drive GP91 FOX as opposed to an ubiquitous promoter. I've told you about gene modulation exploiting RNA technology, so additional trick that you can use is to embed microRNA targeting sequences in your vector so that the RNA is degraded in cells where you don't want the expression to occur. And lastly, gene editing, that I'll come back in a minute to. We're increasingly thinking about using alternative target cells. Of course, the famous one for this is T cells in cancer immunotherapy, but also as the field of somatic cell reprogramming progresses and one can derive transplantable hematopoietic stem cells and probably other cells from those cells, we can envision that this would be an ideal cell to target because it's expandable in vivo, it's clonable, and therefore you can do very serious molecular characterization of those cells prior to the use in a human being. And what's interesting is as we've gotten better at this, we're realizing that there are advantages to having centralized manufacturing facilities. This is particularly true as companies get into this. But if we don't use that process, then we have to be quite rigorous in validation of centers' manufacturing processes. So I just think it's important to end with what the future is going to look like. And I think we're entering an era where therapeutic genome editing is going to be possible at large enough scale and with high enough efficiency that it will be useful in the clinic. And so just the schematic, the cartoon is we would, on the platform of the current gene therapy trials, we would basically collect stem cells, take them to the laboratory, do genome editing, and then reinfuse the cells. And how can we imagine doing that? Well, I think in the first iteration, which is really going to be quick, I think, we would rely upon the high frequency of non-homologous enjoining rather than the high fidelity of homologous recombination. And that difference in the efficiency of those two processes would mean that if you simply want to delete a sequence as opposed to editing a specific mutation, which you would eventually want to do for many diseases, the efficiency is starting to reach the point in large scale where that might work. And we're working on, in collaboration with Dan Bauer, who's a junior faculty in our group, is, again, BCL11A. So Dan has very nicely, this work was recently published in Nature, demonstrated that in BCL11A, there's an enhancer sequence that specifically regulates expression only in red cells. So, in data I won't show you here, if you delete that sequence, you don't affect expression in lymphoid cells and stem cells, but you do affect expression in red cells. And so you can see here, when he compares the level of induction of hemoglobin, fetal hemoglobin, in the purple bar, by deleting the entire gene or exon 2 of the gene to a simple mutation in this enhancer region, you maintain induction of fetal hemoglobin in these cells, shown here, but you have no effect on lymphoid development at all. And so this is a perfect system by which to test the hypothesis of genome editing. And this sort of shows you in schematic form the vision of this. Here's this erythroid enhancer that sits in an intron of BCL11A. And we know that there's natural variants in this enhancer that lead to higher hemoglobin F. This is genome-wide studies. And what we know now experimentally from his work is if you delete this enhancer, you get even more prominent induction of hemoglobin F. And because you're using non-homologous enjoining as an approach rather than homologous enjoining, the efficiency in CD34 cells is starting to get to the point where we can envision this being applied sometime in the near future. So I think this is my final slide. There are a number of trials shown here that are currently ongoing in gene therapy. I think this just, again, shows you how the field is rapidly evolving, and now we would add to this, I think, sickle cell disease, where we will target BCL11A, and again, we hope to have that trial open by early next year. So in there, I did want to recognize people that have helped on the SCID trial. So at Children's in Boston, a really terrific immunologist, Gigi Notarangelo, junior faculty in my division, Sung-Young Pai, who specializes in transplants for immunodeficiency. And we were the sponsor for the US sites, but the overall investigator is someone many of you may know, Adrian Thrasher at Great Ormond Street in London, and we had sites in Paris, at UCLA, and Cincinnati Children's, and collaborators, long-standing great collaborators in Hanover Medical School in Germany. And I mentioned already Rick Bushman did the bioinformatics work, and we were funded by the NIH to do this multi-institutional trial. The BCL11A work is done primarily by two postdocs in the lab, Sarupa Gouda, Christian Brendel, and my longtime lab manager, Chad's actually been with me for 25 years now, if you can believe it. And so he did a lot of this work. And we collaborated with Dan, I mentioned in Stu's lab, Richard Gregory, who's a microRNA specialist up the hallway from us. And we had work, collaborative work with the Broad Institute. So thank you. I'll stop there. And if we have time, ask some questions.
Time for a few questions. David.
I noticed in the LCR vector you changed the orientation of the insert to make it go in the backwards direction. I was just wondering what the rationale for that was. Yeah,
in the other direction, we have adverse effects on titer of the virus, probably due to splicing that occurs in that direction. We're not completely sure yet, but that's what we think is happening. So.
There's been some data that lentiviruses are less prone to integrating near active genes compared to the gamma retroviral vectors. Do you have any data on that from your SCID3 trial and whether that's giving it to?
We haven't done the comparisons ourselves. It's true that there is a statistically significant difference in integrations near transcription start sites for gamma retroviruses versus lentis. That's a theoretical safety advantage, but it is also the case that the very first patient that got a lentivirus in the beta globin trial in Paris, because of the integration away from the transcription start site, developed abnormal splicing and had a clone develop because of that. So I think anytime you have integration, you have some risk involved. But we've changed in SCID3 to lentivirus backbone, oddly enough, not because of safety issues, but because of manufacturing issues. So turns out all of our trials are now lentiviruses, and the GMP protocol for those two things are significantly different. And it just turned out from a manufacturing standpoint, it was easier to do the trial in a lentivirus backbone as opposed to the gamma retrovirus backbone. That was really a practical consideration.
Suzanne?
David, you showed us very convincingly that you didn't have that same clumping of integration sites near proto-oncogenes in SCID2 than SCID1, but I missed why you thought that was the case.
Yeah, probably was, I didn't make the point clear enough. Because when we looked at the pre-infusion product, there is no great difference in integrations. The lack of clumping, or the clumping that we see in vivo in SCID, in the first set of SCID trials, reflects the biology of integrations driving proliferation of cell clones. So it's not as if the integrations themselves were different, it's that the result of the integration biologically was different.
After, say, 3
Yeah.
or 4 years,
Yeah.
rather than at the beginning.
Yeah,
Okay, I misunderstood.
yeah.
Lynn?
How far away are we from stem cell therapies without vectors, using CRISPR-Cas9 to correct genes?
I don't have a crystal ball, but I think it's within. I probably will not be retired yet. I'm getting close. The issue that the people are dealing with, of course, are two. One that we know very well from the retrovirus field, which is the efficiency with which you can do this in cell lines is dramatically higher than the efficiency that you can do this in a stem cell, let alone a reconstituting stem cell. That's a big issue. And the second is, of course, and it will be worked out, is the potential off-target effects of any technology where you're making double-stranded breaks in the DNA. The CRISPR-Cas9 folks swear up and down that there's very few off-target effects, but I think when you're talking about putting lots of clones into a human being, you got to be pretty sure that you're not putting in mutagenic cells. So I think that'll be worked out, and maybe faster than the other issue, which is the efficiency issue.
I was interested in the field where it sits currently with non-integrated lentiviral vectors, because it seems that they had a flourish in the literature and they've kind of petered around.
It's very robust, and I think in Boston alone there must be 3 companies developing, and there's probably more companies than there are patients to be treated. But in retinal diseases, multiple monogenic diseases are now being addressed with AAV vectors. And in hemophilia, it's clear in factor IX deficiency this works.
Sure,
and,
sorry, I wasn't asking about AAV, because that's definitely flavor of the month,
sorry?
flavor of the year.
Yeah.
I was asking about the non-integrating version of lentiviral.
And so what's the question?
Is anyone doing much on that at the moment?
Yeah, but I think my sort of feeling is that the development of that is actually more around delivering parts of the homologous recombination approach in a non-integrating fashion, as opposed to using it as for instance, using it in the brain or using it in hemophilia. There are people developing those for sure, But I think what's attractive about this would be, I think I was talking about this earlier today, but delivering the scissoring mechanism in gene editing with something that doesn't integrate, because you naturally then have gotten rid of potential mutagenic substrate by the cell proliferating. So I think there's a lot of work going on actually in that area.
If there are no last questions, it just remains to again thank David for a wonderful talk.